Welcome to our webinar comparing the BioCyc and KEGG pathway databases. My name is Peter Karp and I direct the Bioinformatics Research Group at SRI, where we produce the BioCyc database and web portal. So that means we of course know BioCyc a lot better than we know KEGG, but we'll do our best to give a informative comparison, although there are a number of places where we'll note where our knowledge of KEGG falls short. So the main criteria for our comparison are going to be the database content of KEGG and BioCyc and the website tools that both systems offer. And we'll be comparing March 2025 versions of both KEGG and BioCyc, where the KEGG data was obtained by downloading it via the KEGG REST API. Now, for a lot of this webinar, we'll be making use of this detailed comparison on this online document, which we will be updating periodically, and we'll go over to this document in a few minutes. And this webinar is being recorded and will be available later on. So first, let's look at what KEGG and BioCyc have in common. Both import thousands of sequenced genomes, analyze them in various ways, for example, by computing metabolic reconstructions for the genomes, and they both manually curate the genomes to some degree. Now, in more detail, when KEGG downloads a genome, KEGG then re-annotates the genome. It changes a number of protein functions in the genome, for example, which BioCyc does not do. We download genomes from RefSeq, and we keep all the predicted functions as they are, except during the curation process. Now, whether it's a good thing to re-annotate the genome or not, I don't know. I have not seen data to bear on the question of whether KEGG's annotation is better than that within RefSeq done by the NCBI prokaryotic annotation pipeline or not. Don't know which is better there. Now, in addition, when each group computes its metabolic reconstructions for the genomes, they use different algorithms developed by both groups, but overall the algorithms are similar in that they recognize known pathways from a reference pathway database. For KEGG, that reference pathway database is called KEGG reference, and for BioCyc, that reference database is called MetaCyc. Now, I've been unable to find any published details on the KEGG pathway prediction algorithm, so really unclear how that works and where it performs well and where it doesn't. The BioCyc pathway prediction algorithm is described in section 3.2 of this online document at arXiv. That is the document describing providing an overall description of the Pathway Tools software, where Pathway Tools is the software package that we've developed that's used to create new pathway genome databases, infer metabolic pathways, and publish those databases on the website. So when you interact with biocyc.org, the website, you're interacting with the Pathway Tools software. So let's jump into the comparison here, and let's start by looking at the number of genomes offered by both databases and websites. BioCyc has about 20,000 genomes, KEGG has about 10,000 genomes, and that ratio is 1.9. BioCyc has 1.9 times as many genomes as BioCyc. Now, BioCyc has 1.9 times as many genomes as KEGG does. Now that, excuse me, that excludes viruses. KEGG does have many viral genomes, and BioCyc does not have viral genomes. We've chosen to focus on bacteria and higher organisms. BioCyc does have 77 curated databases. So of the 20,000, 77 are curated. And those databases have been curated from a total of 153,000 publications. We don't know how to obtain the corresponding numbers for KEGG. Not sure if KEGG has the concept of a curated database versus an uncurated database, or if they kind of curate everything to some degree. don't know. Now here we'll compare the two reference databases, Metacyc versus KEGG reference, along a number of dimensions. Essentially, how many objects of different types do they provide? The first line looks at the number of pathways provided by Metacyc versus KEGG, and here we're comparing the biological pathways, what I think of as kind of atomic pathways that are inherited through evolution as a unit, regulated as a unit, and typically present in their entirety in a single organism. So these are relatively small pathways. They can be bigger than this. Here's an example of the same pathway for cysteine biosynthesis found in both KEGG on the left and BioCyc on the right. So these are highly comparable pathways. Some of these pathways will be significantly larger. They could be up to 10 reactions or 15 reactions, sometimes more. So at this pathway level, Metacyc has 7.1 times as many modules as KEGG does. Now, both databases also contain larger pathways, which Metacyc calls superpathways and which KEGG calls maps. KEGG has more maps than Metacyc has superpathways, so Metacyc has 0.7 times as many superpathways as KEGG has maps. And if we add together those two lines, the pathway and superpathway lines, we'll get the all pathways line. And so overall, if we look at all pathways, Metacyc has 3.5 times as many as KEGG does. If we compare reactions, we'll see that Metacyc has about 19,000 compared to 12,000 in KEGG for a ratio of 1.6. The number of metabolites in the two databases is very similar. Now on that last line, again, we don't know the comparable number for KEGG. What we're counting here is the number of textbook page equivalents found in the mini review summaries that our curators author for, typically mostly for proteins and for pathways. So these are multi-paragraph summaries of what's known about a given gene or pathway that introduce them to someone not familiar with those in detail and provide citations to the literature. So Metacyc, and this is actually a BioCyc-wide number, includes Metacyc and the other databases. They include about 11,000 textbook page equivalents. And it's actually instructive to look at the time course of the number of reactions in both databases over time, because not only does BioCyc have significantly more reactions than KEGG does, but BioCyc is actually growing twice as fast as KEGG is, at least over the last 8 years, where BioCyc has grown by 5,000 reactions, KEGG has grown by about 2,500 reactions. All right, I now want to switch over to that online document that I referred to that actually compares UniProt as well as BioCyc and KEGG, although I'm not going to say any more about UniProt. And this table in this document compares the data content of these databases. And we've been over a number of these numbers already, which I'm not going to go over. So let's take it up at this line, the transcriptional regulatory network line. This is actually a really significant area where BioCyc describes transcriptional regulatory network information on the order of 100 or more regulatory relationships for 11 different organisms. And some of them, such as E. coli and B. subtilis, for example, E. coli in particular is quite extensive, contains thousands and thousands of regulatory interactions, whereas KEGG does not have such information. BioCyc also includes gene essentiality data and phenotype microarray data, so we'll download such data sets and load them into some PGDVs. I believe there are about 30 organisms that we have gene essentiality data for. KEGG does not have those data. We also integrate and this is often through Curation, will integrate enzyme activators, inhibitors, and cofactors, protein localization information, protein features like the positions of active sites and metal ion binding sites, enzyme kinetic parameters. KEGG does provide protein 3D structures, which BioCyc does not. We also integrate gene ontology annotations. Evidence codes are used to label information, such as gene functions, to tell us when a gene function is experimentally determined. And we have these curator-authored mini-reviews and many, many literature citations throughout the database. All right. Let's now look at, actually, I'm going to go back for a minute to this slide, which, yeah, so this slide provides a bit more detail on the information that BioCyc integrates when we build a new pathway genome database. As mentioned, we obtain the genome, the annotated genome from RefSeq, convert it to our pathway genome database format. We then combine computational inferences that we apply to the database with data import operations. Among the computational inferences are that we predict the metabolic reactions catalyzed by the enzymes of the organism. We predict the metabolic pathways that those reactions are part of. And we also compute orthologs between the proteins in the new organism and proteins in existing BioCyc organisms. Now, all the steps in red are also done by KEGG, but the steps in blue are not done by KEGG. So additional computational inferences that we perform are to predict transport reactions that the transporters of the organism catalyze. We predict pathway hole fillers. We predict operons of the organism and its protein complexes. And we compute PFAM domains across the organism. For data import, we go to a number of databases such as Uniprot, and we import regulatory data when it's available, subcellular location information, GO-terms and protein features, as well as organism phenotype and gene essentiality data. And I'll note that not all of this data is available for every genome. It really varies how much of this data is available. But these computational inferences are done for every organism, with the exception, for example, that for the eukaryotes, of course, we don't predict operons. And I'll note that users can request new BioCyc genomes at any time. Now let's go back to this document, and we'll go up to a table now that compares the software tools present in the different websites. And we've organized this comparison into genome informatics tools, pathway informatics tools, additional tools, and omics data analysis tools. So let's go through these in detail. Both KEGG and BioCyc offer linear genome browsers, whereas only BioCyc offers an additional circular genome browser, and BioCyc's linear genome browser can also be used in a comparative mode. And let me give you a sense of some of these different tools and how they work. Okay, so actually let's start with the KEGG genome. So this is the KEGG genome browser, and we're now looking at it applied to the E. coli K12 genome. There's a low resolution view of the entire genome at the top, and we can click on a region here to display any region, that corresponding region of the chromosome. And each rectangle here is a different coding region, and we're looking at kind of a wrapped display here where the lines continue one after the other. We can zoom in and out of this view by using the scale button. So here we're zoomed way out to look at a large region, and here we're going to zoom way in to look at a somewhat smaller region. Okay. Now what I want to note is that as we zoom in, we're not actually seeing any more information. We're still simply seeing rectangles that depict coding regions as well as their directionality within the genome. And there are a few other tools here. So, for example, we can turn on the display of gene symbols. We can also use this box to change the positioning of the diagram. So here we see the BioCyc genome browser, which actually uses the same line wrapping paradigm as the KEGG genome browser does, which actually not many other genome browsers do. And in this diagram, adjacent genes in the same color are in the same operon. KEGG also colors its genes, but I don't know whether these are in the same operon or not. Perhaps they are. We also see in the diagram down here, notice there's a gene with an X through it. That's a pseudogene. Here we see some genes with different looking arrowheads. Those are RNA coding genes. And up here we see this large blue shaded area. That's a cryptic prophage in the E. coli genome. Here's some more pseudogenes over here. And of course, if we mouse over a gene, it identifies what it is. Now I can use the mouse wheel to zoom in on this diagram, and as you can see, it zooms in very fast. And as we zoom in, we do see more information. We see promoters being displayed. We see transcription factor binding sites appearing. That's an ARG-R binding site. We also see terminators are here somewhere. Can't quite spot them, but there's some terminators right there. Here, there are the tRNA coding genes. And if we click here, we'll see the legend that depicts all the different graphical elements that depict, for example, mRNA binding sites and ribosome binding sites. So there's a lot of different sites that appear in the diagram. And in addition, we can zoom into the sequence level, where we see for the RNA coding genes, we see just the 2 DNA strands, but for protein coding genes, we also see the amino acid sequence here. Now, the same tool can be invoked in a comparative mode where the display looks like this. And in comparative mode, the colors indicate orthologs. And what we're seeing here is each line is a single genome. We've asked it to compare 6 different E. coli and Shigella genomes and to align them at orthologs of the ARG-A gene. So essentially we started at the K12 ARG-A gene and then added in alignments of these other genomes. And we can see very quickly that some of the genomes are conserved with respect to K12, like this one, and some genomes are not conserved. There's an insertion or deletion here. And so if we can use the same mouse wheel to zoom in or out. And again, if we zoom all the way in, we should get to the sequence level. Sequence will display eventually. Okay. Let's go back to all right, so that's the genome browser comparison. BioCyc also provides a regulatory network browser that can depict the entire genetic regulatory network of an organism. Not going to spend time on that, but that's also something that KEGG lacks. Now, both BioCyc and KEGG provide information pages for genes and proteins and RNAs. BioCyc, however, provides an additional advanced search tool. Let me show you that real quickly. If we go up to Tools and under the Search menu, look at Search for Genes, Proteins, or RNAs, you'll see that there are different boxes down here that we can open that allow us to specify a range of different search criteria. So for example, if we want to search for all proteins with less than 75 amino acids, we can type in that constraint, submit our query. Here's our result. We get a table of these genes. We can do an ascending sort. We see there are actually 2 genes in E. coli with that are only 8 amino acids in length. Let's go back to this screen. Okay. All right. For some reason that tool is frozen. I'm going to show you that there are similar search tools for pathways, for example, for, okay, and also for metabolites. And we can apply multiple of these tools together. So for example, the metabolite tool lets us search for metabolites by monoisotopic mass, by molecular weight, by chemical formula, by chemical substructure as well. Let's go back to the gene protein page since we didn't quite finish with that. I'll just go through some of these other criteria. We can search by the PI of the product, the protein copy number, and we can search by small molecule regulators or cofactors or substrates or ligands of a given protein. We can also search by its level of characterization. We have a recent paper on this, and by publications cited by the gene. So, and these can be combined. We can select any combination of these, not just one at a time. Okay, now both systems provide a blast search capability. BioCyc provides also a sequence pattern search capability that KEGG lacks, and BioCyc also provides a multiple sequence alignment tool, which KEGG lacks. Both systems allow you to retrieve the amino acid sequence and the nucleotide sequence. For pathway informatics tools, as we just saw, BioCyc has advanced search tools for metabolites, reactions, and pathways that KEGG lacks. Both systems do provide information pages for pathways, reactions, and metabolites, including pathway diagrams in both systems, and both systems provide zoomable metabolic network diagrams. BioCyc provides an interactive tool called the Metabolic Network Explorer that lets the user graphically explore the metabolic network by kind of moving from one reaction to a connected reaction to another connected reaction. Both tools provide what we call a metabolic root search tool that lets the user search for a path of reactions that connect the starting molecule to an ending metabolite. And then there are a number of additional tools that are provided by BioCyc that are not in KEGG. The ability to visualize a number of types of regulatory network information, the ability to visualize the regulon of a transcription factor. So for example, if we go to a transcription factor page like TRPR, and if we click on, so this is the TRPR gene page, if we click on regulon, we'll see all the operons that the TRPR transcription factor controls. So we see diagrams for all of them. There's also an extensive facility in BioCyc called Smart Tables that enables the user to manipulate lists of metabolites or genes or pathways or binding sites, store those lists in your BioCyc account, analyze them. For example, given a gene list, you can transform that to a list of all the metabolic pathways containing those genes. And there's a really extensive comparative analysis tool suite. So under the Tools menu, if you select Comparative Analysis on the far right, you'll see many built-in comparative tools. Now this next part of the table compares the Omics data analysis tools present in the two systems. And we've broken it down by transcriptomics data analysis and metabolomics data analysis. And you'll see that a number of the tools are shared between both types. For example, we're able to, in BioCyc, we can perform enrichment analysis on both metabolomics data and transcriptomics data. And we can paint both types of data onto individual pathway diagrams, which KEGG will do as well. And we can paint both types of data onto a zoomable metabolic network diagram, which KEGG can do as well. Now, what KEGG cannot do, I believe, is it cannot animate these displays. So, for example, should have an example of this here. Yes. So this is the E. coli metabolic network diagram painted with transcriptomics data. The color scale is shown over here. And so we've inputted a data file where each line of the data file describes one gene and lists multiple data columns that give transcriptomics abundance measurements for that gene at different time points in the experiment. So there are 6 different time points. Now, if we click on one of these reactions, we can bring up what we call an Omics pop-up that graphs the expression levels of this gene over the 6 time points in this experiment. There are actually 2 genes behind that reaction. And then, as I mentioned, this display can be animated. So this is the animation control. If I click the play button, you'll see that we're iterating across the 6 different time points, and that the colors in the diagram are changing, for example, here in the TCA cycle, as the diagram progresses through the different time points. And this is a data set that depicts the transition of E. coli from anaerobic to aerobic growth. So you can see the TCA cycle turning on as we get to the aerobic phase. And we can also step this animation 1 frame at a time. We have a few additional tools on the metabolomics side. One additional shared tool is called the Omics dashboard, which I'm not going to take time to describe today, but it's a really different, unique way of visualizing the entire cell and its transcriptomics or metabolomics response. We have some, as I say, additional tools for metabolomics data analysis, one example being the ability to compute the set of pathways that will cover or account for a metabolite set. All right, let's now go back to this other presentation. And let's talk for a few minutes about the curation that both groups do. We don't have a good sense of what KEGG's curation strategy is. We don't have a good sense of how many total articles they've curated. What's their curation strategy? Is it per organism or per protein family, for example? We don't know. How do they prioritize their curation? How do we find curated entries within KEGG and recognize that a given entry has been curated? And which organisms in KEGG have been curated? So if anyone knows the answer to those questions, be quite curious to find out. Now for BioCyc, essentially when we curate a new database, we typically will take on curation of one organism at a time and do a really in-depth curation of it, where the first stage is reviewing computational inferences done by BioCyc when the database is created. For example, we'll review the pathway predictions for a given organism, and the curators will look for pathways that may be in error. For example, if the system predicted the presence of a photosynthetic pathway in an organism that doesn't do photosynthesis, that pathway would be deleted. Similarly, we review the protein complex predictions, etc. We then do literature-based curation. We'll do a literature search for that organism and identify articles that typically define new molecular results. We're not interested in clinical articles. That's not part of our curation. So for example, we give high priority to articles that have new gene functions or publish new pathways. And the curators will author the mini review summaries for genes and pathways. They'll add evidence codes that let us, so for example, here's a gene page for an E. coli gene, TRPA, and it has an evidence code, this Flask icon, that tells us there is experimental evidence for the function of this gene, and it comes from these papers here. The curators will also update various database fields like gene names and synonyms, enzyme cofactors and activators and inhibitors, and define regulatory information like transcription factor binding sites and promoter locations. We also sometimes import this data from other sources. So if you're aware for an organism of interest to you of, say, a promoter data set or a terminator data set that you'd like us to import, contact us and let us know. Here are some of the databases that we've curated in BioCyc. We have a number of curated databases for the eukaryotic model organisms, and we have a human curated database. We have curated databases for a number of bacteria, including model bacteria, human microbiome members, and various pathogens. We also have 6 curated databases for cyanobacteria, as well as curated databases for all the highly used synthetic biology chassis organisms. Now, you might be curious to know, how much curation does BioCyc actually do? And we were curious about this as well because we didn't know the answer to that question. So we've done a case study on Cupriovetus necator, which is a synthetic biology chassis organism. And the first thing we did was to issue database queries that asked how many proteins in this database have changed their name, their function, since the database was created. And really the only reason that would happen would be if the protein was curated. And the answer to that is 858 proteins had their function changed. Now, as we thought about this more, we realized that probably not all of those changes are significant. So for example, and here's an example of an insignificant change, is where the same transporter function was rewritten in a different way without really changing its meaning, but perhaps in a more standardized way. So this is another thing our curators will do, is to try to rewrite protein functions to use standard protein naming conventions, such as those developed by Uniprot and by NCBI, because we try to take advantage of use those same naming conventions. And although that is a useful, helpful change that has value to users, it's not the same as changing the function of a protein from a vague function that simply places it in a protein family to a function provided by a paper that we encountered that actually gives a molecular function, an exact enzyme function to this protein. So to determine how many of the 858 were likely to be significant changes, we selected 200 of those 858 proteins at random and reviewed them to find which were significant and which were not significant. And we found that 93 of them, of the 200, were significant. So if we extrapolate that rate of 46.5% to the 858, we see that 399 of the 858 are probably significant annotation changes, which in my view is a large number. That's a good fraction of the genome that's been updated by our curation procedures and improved to give more accurate annotations. Now, we've actually done that same study across 10 other genomes. So we've chosen 200 proteins at random from the list of proteins that had changed annotations. For a total of a bit more than 2,000 proteins have been surveyed in this way. And what we found was that on average, it's about 50% of the proteins with changed annotations had significant changes. And overall, about 311 proteins is the average across all these different genomes for how many proteins had significant annotation changes. I'd now like to talk for a moment about the KEGG and BioCyc approaches to visualization of whole organism metabolic networks. And so we're going to take a look at the BioCyc and KEGG cellular overview diagrams, their whole metabolic network diagrams, and I want to raise the distinction of the approach of creating organism-specific metabolic network diagrams versus what I call Uber charts or Uber network diagrams. The difference being for BioCyc on the left, our metabolic network diagrams reflect one organism. So each diagram contains only those reactions and pathways that are present in a single organism. Whereas in KEGG, they're combining reactions and pathways for many different organisms onto the same diagram. So that can be useful. It can be useful to combine them into one diagram if you want to be able to see, for example, what fraction of all known pathways are present in a given organism. But creating these kinds of Uber charts, well, another advantage of creating the Uber charts is that you only have to draw them once. Okay, so, or well, as your reference pathway database expands, you do have to add new pathways to it, but you don't need to manually draw the diagram for every different organism in your collection. Now in BioCyc, we don't manually draw them for each organism. We have algorithms that create these diagrams for each of the 20,000 organisms in BioCyc. Now, one possible advantage of organism-specific diagrams is they can be viewed as more accurate because they depict exactly the set of pathways and reactions in one organism, and they don't include additional pathways. They're also simpler in that, for example, E. coli has roughly 400 metabolic pathways, and we're including only those 400 as opposed to the other 2,900 pathways across Metacyc. Because remember, Metacyc has about 3,000 pathways. So E. coli only contains about 10% of the universe of metabolic pathways. And so creating diagrams that contain that full universe, they get very crowded. That can be a disadvantage of that approach is the crowding that goes on. Now, I'd like to indicate the structure of the BioCyc diagrams. They're all laid out using the same basic approach, which is to put catabolic pathways on the right side, biosynthetic pathways on the left side, energy pathways down the middle, and to have a plasma membrane and a cell wall and periplasmic reactions and extracellular reactions. These are reactions that occur in the cytoplasm but are not part of any particular pathway. And then we organize the pathways into sub-blocks by grouping together related pathways. Like these are all the nucleoside and nucleotide biosynthesis pathways and the amino acid biosynthetic pathways. Okay, so this is the human cellular overview, and of course, since humans have a different complement of pathways, it has a different shape than the E. coli diagram, but again uses the same basic layout where the biosynthetic pathways are on the left. Now, with BioCyc diagrams, we have semantic zooming as we use the mouse wheel to zoom in on a given pathway, and let me show you that real quickly. If we go here, we can zoom in on any region by flicking the mouse wheel, and as we zoom in, we see more and more information. We see metabolite names appearing, and we see gene names and enzyme names appearing as well. We can also do highlights on the diagram. So here we've highlighted in red every reaction whose genes are regulated by the transcriptional regulator FNR. We've also created cell type specific versions of the charts. For example, in humans, here we're showing the human epithelial cell diagram by graying out all pathways not known to be expressed in epithelial cells. And we can also put up multiple charts for organism communities. So here we're looking at 8 different metabolic charts simultaneously for a simplified mouse gut microbiome community. And so each chart is for a different organism. The gold pathways are those that are specific to a particular organism. And we're also showing connections where a secreted metabolite is taken up by another organism. And as you've seen before, we can overlay transcriptomics data and metabolomics data on this chart. We can also put on multi-omics data. So more recently, we've added the capability of showing 4 types of omics data simultaneously using coloring and thickness of reaction arrows, such as shown here. Notice that both the coloring and the thickness are changing. And we can modify the coloring and the thickness of the metabolite nodes. So those change as well. So let me summarize the comparison that we've performed, which has shown that BioCyc has more data than KEGG, about almost twice as many genomes, 7 times as many base level metabolic pathways, 1.6 times as many reactions. And these differences have real implications for some of the algorithms that make use of these data. So for example, metabolic reconstruction works by recognizing known pathways from a reference database. So if one reference database is larger than the other, it allows the algorithms to recognize more pathways. Similarly, if we're doing metabolic root searching, pathway design, we have a larger palette of reactions to design new pathways from, and BioCyc has a larger set of reactions to do gap filling of metabolic models. We also saw that BioCyc adds more information to each genome by doing both data imports and computational inferences. So it assembles an organism database that has a wider repertoire of data than does KEGG. Examples being gene essentiality data and protein feature data. Okay, so that's really the same point here. We also saw that BioCyc has many more informatics tools than KEGG does, from the circular genome browser, the comparative genome browser, and the ability to animate omics data displays on metabolic network diagrams. We've also seen that KEGG has many unknowns, that there's not a published description of its pathway inference algorithm or its curation strategy, which makes it hard to understand how KEGG is being created. Let me skip ahead to a few final slides here. We'd like to encourage you to consider switching to BioCyc. BioCyc is subscription-based, and you can find out more about our subscriptions here. And you can contact our sales group here. We're making a coupon available to you all that's good through the end of September and gives you 50% off the price of a BioCyc subscription. Here's a publication that will tell you more about BioCyc. We do have video tutorials on many aspects of BioCyc, how to use it. We have in-depth website help, and again, the same publication. So thank you for your attention.